All right, we're going to start the lightning talks. You guys are going to get to hear some of the really cool stuff that your fellow attendees are doing with PACE data and why they're interested in PACE data. I'm going to hit the play button, and then they will each have 2 minutes to tell you about themselves and their work. So let's go ahead and, all right. I'm gonna hit go.
Good morning, everyone. We are from the University of Rhode Island. I'm Ali Akanda. I'm a hydrologist interested in water quality monitoring. Retno is my PhD student. She's here somewhere. And Colin is our colleague from the Graduate School of Oceanography in Rhode Island. So we are interested in water quality monitoring, both freshwater, inland freshwater, as well as coastal water quality monitoring. We are looking at more at the vulnerable delta regions across the world, like the Bengal Delta, the Mekong Delta, as well as on Borneo Island, where actually there are a couple of very interesting delta ecosystems. Why are we interested in PACE? Of course, the very excited about the increased spatial resolution, the spectral resolution. We are very much interested in the seasonal variations in water quality because there is a lot of seasonal monsoon and freshwater flow in these ecosystems, and we are hoping that with PACE we will be able to capture the variations in water quality. And the reason we are interested in these in this is the applications in, we are very much interested in the applications in public health. We work a lot in South Asia and Southeast Asia with public health partners, and we are very interested whether we can use PACE data as a new way to strengthen the early warning systems and more informed community-based decisions to empower the actual our partners in those parts of the world.
Is it not changing?
Say that again.
Is it not changing?
Oh, not changing, no. Thank you. So I'm here for the whole day. Please catch me.
Oh, there it went.
Oh, nice.
That was a long 2 minutes. I watched this whole thing run. It works.
Thank you.
Okay, good morning, everyone. My name is Kamal Arial from University of Maryland, Baltimore County. As you can see from my title, I'm interested in aerosol and ocean color. So there was a question in the icebreaker, rank your interest. So I ranked aerosol first and then ocean color second. And I was hoping aerosol should come second. It went to third option, but that's okay. But anyone interested in ocean color, I think we should have aerosol in the second order because we need to understand aerosol and remove them to get to the ocean color. That's my understanding, and that's what I do. So I'm a PhD student, and my project is to do joint retrieval of aerosol and ocean color using multi-angle polarimeters. So I just had this screenshot of my recently published paper. So we just use polarimetric data, basically HARP2 and SPEX1 from PACE to do both aerosol and ocean color retrieval. And this algorithm is suitable to be used over complex coastal water scene where we have complex aerosols as well. I don't have the plots here because I will show the plots tomorrow. So I have poster tomorrow morning at in light scattering and radiative transposition. And I also have the talk on Tuesday afternoon in the Earth through the PACE lens. So I'll show my work there in detail. Thank you, everyone.
Hello. A very good morning, everyone. My name is Akash Ashapure, and I'm a research scientist at Science Systems and Applications, Inc. I'm here to talk about aqua versus atmospheric correction of multi- and hyperspectral satellite observations, particularly over fresh and coastal waters. To talk about the summary, at Freshwater Sensing Program, we use advanced machine learning techniques like mixture density networks to improve atmospheric correction and water quality products for hyperspectral as well as multispectral images. I take care of the atmospheric correction part, and my colleagues take care of the downstream products estimation as well as uncertainty estimation. Now, why PACE? Because PACE's hyperspectral resolution enhances surface reflectances accuracy, especially in challenging blue bands. And talking about the social relevance, improved water quality products support sustainable maintenance of the aquatic ecosystem. Who are going to be the end users? It can be aquatic researchers, environmental agencies, to name a few. On the left-hand side, there is a screenshot. It's a comparison of Aquaverse and Eltogen surface reflectances in blue bands in 410 and 443. The image was acquired over Lake Erie, captured on 31st of May this year. We see that there are some linear artifacts in Eltogen and also some uncertain regions towards the edges, and we were successfully able to kind of get rid of that, and the results look much smoother. If you're further interested in my results, Aquaverse results, you can find me at AGU on December 11th. My presentation is at 10:30 in the session GC 32A. Thank you so much.
Good morning, everyone. So I'm Zaid Bakri. I'm a PhD student at the Atmospheric Sciences program at Michigan Tech. And my research is about polarimetric measurements of cloud droplet size distributions generated in a cloud chamber. So the objective of, or the goal of my research is to measure the cloud droplet size distributions in a cloud chamber and under controlled known environment. To study or to understand the influence of the microphysical processes that often happen in the, up in the atmosphere in real clouds, but in a cloud chamber at a smaller scale. So why do we choose PACE? So as we know, PACE HARP2 provide us with data on the cloud droplet size distributions from real clouds at larger scales. And this will help us to bridge the gap between the controlled experiments and the real atmospheres, and also to see or look for similarities between large scales and smaller scales. So the societal relevance is to enhance our understanding of the clouds that play a major role in regulating the Earth's atmosphere and the hydrological cycle. So here we have a sample of our theoretical simulations of the polarized light scattered by the cloud droplets, mainly at the 145 degrees. And each of these panels is for the polarized light as a function of the wavelength, and for like different size distributions with a specific effective radius or the mean of the size distribution, but for different widths of the size distribution, or we call it the effective variance. And we mainly focus on the three spectral bands, the red and green and blue, and as we see, we are able to tell the difference between the different size distributions, and from there we know what we are seeing. So I can explain later when we meet. Thank you.
Hello, I'm Ileana, and I'm a postdoc in the Water and Ecosystems group at NASA Jet Propulsion Lab. And I'm using PACE data and other NASA data to look at this issue of wastewater pollution in coastal environments. And so I'm working with some professors and students at San Diego State University to particularly look at the sewage plumes in the Tijuana River, which is the result of some failing infrastructure on both the Mexico and the US side. And so we're really trying to leverage PACE's hyperspectral abilities to just make some correlations and connections and associations between the satellite data and in situ data. And I'm particularly using fecal indicator bacteria and also some sequencing data, which is able to resolve phytoplankton as well as other pathogenic bacteria. And I think there's also some synergies with other hyperspectral instruments such as EMIT. And I think that this is going to lead to just a greater understanding of biodiversity changes as a result of these wastewater inputs, and hopefully trying to connect it more with public health. And I know that the San Diego State University folks are working with the Tijuana River National Estuary Research Reserve and with the California Department of Public Health. And if you want to hear more about this, I have an oral talk on this topic on Friday morning at the NASA One Health oral session. Yeah, thank you.
Hi, everyone. My name is Lily Dove. I am the second Rhode Islander up here this morning. We are the smallest state, but we are also the ocean state, but I'm a postdoc at Brown University, and I am a physical oceanographer by trade, but I am exceptionally interested in PACE because it offers us the ability to confirm some things we've expected to be true in numerical models for a long time, but now we're actually able to observe them. So with SWOT, which is the Surface Water Ocean Topography Instrument, we are able to find density fronts in the surface ocean. These are places where we expect there to be enhanced exchange between the surface and the interior, which can bring up excess nutrients into the surface ocean, which then go on to form phytoplankton blooms. And we have seen in numerical models that regions with fronts have greater plankton diversity as opposed to regions that don't. Combining SWOT and PACE data will allow us to confirm this globally. My special interest is in the Southern Ocean, but I'm interested in expanding outwards. And this could be of a special interest to the fisheries communities as well as those interested in water quality as well. You can find me at AGU. I'm giving a presentation on Monday afternoon. And I will also have a poster on Friday morning. I look forward to talking to all of you. Thank you.
All right, perfect. Good morning, everyone. My name is Robert Emberson. I'm a researcher at NASA Goddard and UMBC. I'm actually a surface process scientist thinking about erosion more than anything else. And many of you might be aware that soil erosion of agricultural and topsoil elsewhere can drive both eutrophication and siltation of river systems and reservoirs and offshore. So for the last 2 years, through other work, I've been developing a global soil erosion dynamic model, and using this, I can estimate soil erosion kind of at 1 kilometer grid scale every month around the world. And so this has been a really helpful model. Building with, we've used this to think about kind of impacts on hydrological systems. And my student assistant Pranali, who's here today, has been doing a great job of working on analysis of, in this case, global reservoirs. And we've thinking about 168 global reservoirs of mid-size and looking at Landsat and DCI data. And so this is actually not PACE data yet, but it's really interesting to think about this because we've seen strong correlations in many parts of the world between the erosion happening upstream and the impacts on chlorophyll in those reservoir systems. So this is really suggestive of strong connections between potentially the erosion processes and the impacts on biochemical cycles, but we really don't know much more than that from this sort of very coarse Landsat data, and the connections to these biochemical cycles are really up to be kind of analyzed more. And this is why I'm super excited about PACE data. I'm going to start playing with it to think about how can we understand more about what the impacts of erosional processes, particularly the impact of fertilizer from agricultural soil, driving, and the timings of that, and how it drives the change in HAB formation and things like that. So happy to talk more about this. I have a presentation at AGU this week on the soil erosion elements and the impacts on reservoirs, but I'm really, really excited to think more about how we can do more with PACE to think about erosion and downstream impacts. So thank you.
Yeah.
It's this one.
No, this one. Yeah, this one. Yeah, that's the correct one. Hi, my name is Chandra Gangodakamge, and I'm a PI for the
NASA Water Resources Program project for the Congo River Basin. So what we primarily do is like we use the NASA data to quantify the reach, scale, all remote sensing parameters, surface water extent, and discharges, etc. So in this figure, you can see that like we are primarily interested in quantifying the dissolved organic carbon content in river reaches, especially in the Congo River Basin. You can see that the main river is going around the basin. It's not really crossing the basin, and it's going close to the river ridge line. And all tributaries are joining to the main river from the left-hand side. And all the forests are located within the central part of the basin, and the water primarily joining, we call it a black water. We want to quantify the organic carbon content in the river ranges. So I think that this PACE data set is a very good data set, specifically because the river is large, and this part of the river, it is like 8 kilometers in the width. So we are hoping to quantify, use the PACE data set, specifically quantifying the organic carbonic content, and make this data set available for the river reach scale. So other than that, we have other data sets like salt data sets, water surface extent, and all these data sets are available, and we quantify it and make it available. And so once we figure out how to do the quantification for the organic carbon extent, so we will make this data set available for the community.
Hello, I'm So Young Ha from NCAR. I'm a PI of this PACE project titled Advanced Air Quality Forecasting through Coupled Data Assimilation of Hyperspectral PACE Aerosol Retrievals and All-Sky Radiances from multiple satellite sensors. So we are building NSF NCAR's next generation air quality analysis and forecasting system to assimilate PACE AOD in JEDI for MPAS Scorecard, which is our new line-coupled atmosphere aerosol model using variable resolution capabilities for both regional and global settings. Why PACE? We, as a data assimilation people, are always hungry for more good data sets, and we are trying to go beyond monitoring air quality. By assimilating the PACE products, we are trying to improve our air quality forecasting. Through that, we believe that we can better serve the community by protecting public health and ecosystem. So we are going to, once we build this system, we are actually in the work in progress. Then once we build that, we are going to test this new system operationally over India. And then by the end of the project, we are going to release it to the community. So as you can see in the diagram below, with all the color boxes indicating all the development and improvement we are making for PACE data assimilation, AOD assimilation. But once it's built, then it's easily applicable to other, satellite products as well. So this is a highly interdisciplinary work, and our team is working really hard to build it, and then soon we hope to release it, to the community at some point. Thank you.
Hi everyone, my name is Karm. I am an undergrad student that worked at the Lawrence Berkeley lab. I was working on a subglacial hydrology model, which is water under ice sheets. Specifically, we were looking at Greenland because the region in Greenland, especially in the western coast, is more turbulent there. So there is more activity going on within the water currents underneath. My job during the internship was to develop pre-processing tools to gather parameters such as bed elevation, ice thickness, as well as geothermal flux, and get this data from field data such as NASA Bed Machine and put it inside the simulation so that our simulation, the subglacial hydrology model, will output more realistic outputs. The societal relevance of this is with more accurate subglacial hydrology simulations, it also comes with more accurate ice sheet models. And essentially, accurate simulations in ice sheet modeling can help us prepare and adapt to rising sea levels and other effects from ice melt. The QR code leads to my poster that I presented at the CSA, the Computer Sciences Division presentation, but I am also presenting at AGU on December 11th, sessions C31D, and my poster number is 361 as part of the cryogenic division. Thank you.
Oh, do I have to change it? Yeah, just press the right arrow. Did we get that? Hey. All right. Hi, I'm Eli Holmes. I'm from NOAA Fisheries. For the last 4 years, I've been really involved in Hack Weeks as a way to onboard research communities, scientific research communities, to new data streams, particularly remote sensing data streams, such as the PACE data. So this is in a field of work of trying to accelerate innovation within research communities. When I say Hack Week, you might think, oh, that's a social coding thing. But actually, in this context, a Hack Week is just the culminating event of a much larger thing that involves 3 big components. One is soft infrastructure. These are tutorials targeted towards your research community, packages that really make it really easy for them to access the data. You're lowering all these barriers, but also open computing infrastructure that lowers the barrier to just jump on, get their work done. They don't have to install anything. So it's this combination of these 3 things. The Hack Week is the culminating event. One example of this, or 2 examples, are the PACE Hack Week that happened this August, and then I also was involved with the Ocean Hack Week. And here's an example of how this can accelerate things. So during PACE Hack Week, they had a bunch of tutorials, and then in the period of 2.5 days, a team that I was mentoring and leading developed like 8 new tutorials in 2.5 days, complete tutorials. They made this online book for their particular research field to help people in their field access the data and do something interesting. Here's an example of something comparing chlorophyll products across different remote sensing types. 2 weeks later, I went to Ocean Hack Week with a group and who had never been exposed to PACE data, but I was able to show them the tutorials that were developed for the PACE Hack Week and then also during the PACE Hack Week. And they, again, in 2.5 days, were able to get some pilot ideas going. One project in particular I thought was pretty amazing, got that research paper going in a month. So that's acceleration.
There's a person who's missing. Okay, just toggle. Okay. Here we go. Yep, that's me. You there? All right, so good morning. I'm Maria Kavanaugh from Oregon State University. So MBON, or the Marine Biodiversity Observation Network, is an interagency sponsored effort to establish baselines of marine biodiversity, expand technological capacity of existing monitoring and management efforts, and integrate marine life observations into national and international ocean observing systems. In the Pacific Northwest node, of which I'm a part, we're partnering with NOAA Fisheries, sanctuaries, tribes, state agencies, and others to increase understanding of patterns, trends, and ecosystem functioning, as well as operationalize observations of multitrophic level plankton community composition in the Northern California Current, which is an ecologically and culturally unique area in the California Current. So we embed ship-based observations into dynamic seascape habitats to extend inference in space and time and account for diverse optical water masses, seascapes together with high-resolution multitrophic level imaging and hyperspectral IOPs, aligning with PACE science and applications in a few ways. So we provide a test bed for plankton community composition and size distribution algorithms that are robust across regional coastal seascapes and diverse optical inputs. And secondly, we provide a conduit for PACE-informed data to be ingested into ongoing and developing indicators to inform fisheries and ecosystem management, including things like salmon survivorship and harmful algal blooms. So our project is a new PACE Early Adopters Applications project. So come chat with me. I'd love to learn from you and learn more. And see session B32 for more seascape applications. That's on Wednesday morning.
Looks like somebody else isn't here. Oh.
Hi, my name is Xiaofeng Li. I'm from the Institute of Oceanography. Today, my talk is about how to turn MODIS into PACE. So it's a spectral super resolution of MODIS data for generating PACE-like spectrum. So as we know, the MODIS only have like a few spectrum, and the PACE has a hyperspectral. So what we did is we collect the data in between, and then made a model, deep learning model, to map the relationship between the two instruments. And then after you learn the instrument's characteristics, you can project the MODIS data from right now to 20 years ago. So the key point is to build a hyperspectral MODIS data. So to do that, like any deep learning model, you need basically 4 things. Number one, you need a huge data set. In this case, we have like a 60 million profile matched together between PACE and MODIS. And then the second, you need a deep learning model. In this case, we propagation, fully connected numeric model, neural network. And then the third one, you need, once you have the model, you also need a good training strategy. In this case, we build a nice GAN network, and then you put the information flow correctly. And the last one, you need a huge computing power. So after all that, you can start turning MODIS into PACE. So the upper right corner shows the PACE spectrum. We classify them into 5 different groups using the k-mean clustering algorithm. And for each of these clusters, we use this AI model to do that. So once you have that, you can have a good mapping between the two. Thank you.
Hi everyone, my name's Ryan O'Shea. I'm a research scientist at Science Systems and Applications, Inc. Today I'll be talking about fresh and coastal water product refinement and extension for PACE. This is all part of Aquaverse, which Akash talked about earlier. The summary of our work is we're improving harmful algal bloom monitoring for global inland and coastal waters. We're using PACE due to the hyperspectral resolution, which enables the identification of phytoplankton community composition, in this case via retrieval of inherent optical properties and biogeochemical parameters, thereby improving HAB monitoring. The societal relevance is that the improved HAB monitoring increases inland and coastal water resource managers' abilities to direct limited resources to better protect public health. On the figure on the right, you can see our end-to-end processing framework. It starts with atmospheric correction, which Akash talked about earlier, where we go from the TOA radiance to remote sensing reflectance. We specifically developed a model for inland and coastal waters, trained on simulated data. Akash then passes this remote sensing reflectance to me. I do inversion, generating inherent optical properties and biogeochemical parameters, and eventually phytoplankton community composition. This again was trained specifically for inland and coastal waters, leveraging a large in situ data set of about 7,500 measurements. Both Akash and I then pass our products on to Arun, who's also here, and he generates uncertainty products, which give sort of estimates of how confident our algorithms are in their own predictions for a given region. If you're interested in these models for PACE or models For other multi and hyperspectral instruments, we have a set of the Jupyter Hubs tutorials in the bottom right shown as a QR code. And if you'd like to chat more about any of these results, all of us have posters at AGU. Mine's on Monday and Tuesday. Thanks.
Morning. My name's Francesca. Is that getting back? My name's Francesca Samsel. I'm at the Texas Advanced Computing Center at UT Austin, and I'm a visualization researcher, and I work on large climate models, but specifically multivariate, time-varying models that combine climate systems, so coupled climate models. And what you're looking at here, just this is context for why I'm interested in PACE, is on the upper right, it's MPAS data that has ocean temperature, sea ice coverage, atmospheric winds, and vapor content quantities over time. And so that's where my foundation is, but we have found that a lot of people who have like biogeochemistry data, they need to co-locate variables in a three-dimensional space over time. Okay, so the next image down, those are 4 types of water that are underneath the Ronnie Filster ice sheet, and the scientists want to see how they interacted and moved back and forth. So our goal is how do we help you see more variables interactively so you can better do and see the connections in your science. I also have the task of public outreach. I'm trained as an artist. I have a technical team behind me, don't get me wrong. And so that's what you see on the left-hand side is we are looking to expand the vocabulary which we use to encode scientific visualization data or scientific data so that it's one, more intuitive, but 2, you can put more variables in without having the cognitive load. And I think our website's at the bottom. We're talking on Friday, 2/20, but we do a ton of work, so look at the website.
Okay. Hi, everyone. My name is Samantha Smith. I'm an early career contractor at NASA Goddard in the Applied Sciences Department. I'm pretty interested in how PACE and some other hyperspectral satellites can be used to provide more information in coastal areas and the land-water interface, where data can be pretty sparse and difficult to interpret due to land adjacency effects, especially in the context of providing more information to resource managers so they can make more targeted point source observations and monitor the health of the water bodies that they're interested in. So at AGU, there will be 2 posters that include some of the results I've done using PACE. The first will be Monday morning. I'll be presenting at the air quality session for AeroNet, and it includes some comparisons of PACE as well as some other satellite sensors with AeroNet OC, which is an ocean-mounted radiometer. So here you can see some of those results that compare PACE with OLCI, VIIRS, and AeroNet during different hydrological conditions in the Chesapeake Bay. The second will be on Tuesday. It'll be presented by Stephanie Scholar-Ouz, who's the Applied Sciences Manager at Goddard, and it will include some early results using an inversion algorithm phytotax and applying it to PACE to determine what might be the dominant phytoplankton groups based off of that hyperspectral data. So here you can also see some of those preliminary results where we've used phytotax to predict the dominant phytoplankton groups in the Chesapeake Bay. And we've been able to predict an algal bloom that happened this past summer following some abnormally hot days in June. So thank you all for listening, and I hope to maybe chat with you or see you all at AGU.
This one? No? Okay, thank you. Okay, good morning. My name is Dwi Susanto. I'm a senior scientist at University of Maryland. I'm working in the exotic place of Indonesian seas, so it's basically down there because it's 12 hours different, so it's exactly down there. So Indonesia is very unique because it's the only connection between the Pacific to the Indian Ocean, and then also a center of atmospheric convection. So if you are interested in the cloud, will be very nice. if you're interested in the ocean, this is the only connection from the Pacific to the Indian Ocean tropical region. And also, this is a region where most marine biodiversity in the world, so that it is very important for the plankton. And also, this is one of the center of the Indian Ocean dipole, so that way strongly affected by the ENSO and Indian Ocean dipole. And currently, I have the ongoing project. They call it TRIUMPH, make sure I'm correct spelling. Yes, TRIUMPH. Okay, so it's True Flow, Indonesian Seas, Upwelling and Mixing Physics. So I'm the lead PI. 2 months ago, I deployed 5 mooring. That will be good for the next 3 years with the 1.5 year recovery and redeployment. So this is a collaboration with the Indonesian scientists and also the First Institute Oceanography from China. So this is will be multidisciplinary from the physical, biological, geophysical, and even actually engineering also. So in conclusion, basically that the Indonesian seas is ideal place for the actually PACE validation and also collaboration. So that way for the NASA scientists, the most important is money, money, money. And for the fellow scientists, let's collaborate. Thank you.
Hey folks, Ryan Vandermeulen, NOAA Fisheries. So fish are no doubt using their visual systems for various aspects, including navigation. They're trying to catch prey. They're trying to avoid being eaten. And over time, they can become visually adapted to their optical environment. And there's 2 aspects of this. There's luminosity, the total amount of light, which affects essentially the sensitivity of a fish eye. And then there's also the chromaticity, which is essentially the color sensitivity of that. So what you're looking at here is a 2D interpolation of using PACE data essentially to reconstruct the subsurface light field, not only propagating downwelling irradiance to the depth, time, and place that we're catching certain fish species. And it's not just the total amount of light, but again, that color aspect as well. And you can find the areas that are yellow are highlighting these optical niches where you're more likely to find certain types of fish species. What's really cool is you could actually measure the spectral response functions of a fisheye through a process called electroretinography, and it actually backs up the sort of things you could see from PACE, and you can see that their eyes are adapted to be more blue shifted or to be more sensitive if they're deeper species. If you're a squid, for instance, this graph would highlight in the bottom right-hand corner, showing deeper, more blue-shifted waters where squid are. Fisheries typically doesn't use optics as a predictive measure, but when we do put it into species distribution models, it actually does a bang-up job explaining pretty much the most variance behind temperature as a predictor. So it's very useful. Feel free to come talk to me here. I'll also be at AGU. I won't be talking about this, but talking about GeoXO, which is PACE in a geostationary orbit. So.
Good morning, everyone. So I'm Christopher Anjohi from graduate student at Catholic University of America and a colleague of Google Nairobi. So my research interest in floods events at the Kipini, where Tana River and Indian Ocean meets, and 3 kilometers away during flood events, which happened from March 2024 to May 2024. So I'm trying to monitor the impact of floods on water quality and aquatic life at the confluence and at 3 kilometers away into the ocean during that period. Why PACE? The reason for my interest in PACE is for its hyperspectral resolution and in using chlorophyll A and detecting phytoplankton, which help in detecting changes in water quality and ecosystem health due to flood events. So this will help the the community, which have the water level, the sea level has been increasing over a decade now, which have been displacing people in the near the conference now, which will help now in disaster preparedness and local authorities in at Tana River County, that's in Kenya. Yeah, so as you can see on the bottom right, you can see that's the Indian Ocean towards the left, the far right, and then Tana River entering into the Indian Ocean. So that's where my focus research is in. Yeah, so thank you so much. Happy to answer questions later. Thank you.
Hi everyone, I'm Jake Weiss. I came here all the way from Hobart, where I'm at the Institute for Marine and Arctic Studies of the University of Tasmania. I'm working in Arctic biological oceanography, and I'm looking at factors driving productivity in polynyas down there. I'm using biooptical observation from space, float, and animal-borne sensors. And I've worked a lot with multispectral ocean color observations. I'm not currently an active, but a prospective user of PACE data. And I'm going on a marine campaign, the Demo Marine Voyage, next year in March to the Shackleton Ice Shelf, which is in Eastern Antarctica. And we're aiming to characterize the biogeochemistry and all things phytoplankton down there. We're deploying some BGC Argo floats and seal tag sensors. And on one of the Argo floats, we have a hyperspectral radiometer, the Ramses sensor, and an underwater vision profiler, UVP, looking at particle size spectra. And I think links to PACE I'm hoping to explore are looking at light attenuation from the Ramses sensor compared to PACE observations, and then phytoplankton absorption, pigments, and community composition from water samples, and HPLC samples and absorption observations, and then also carbon export, which we can get from the UVP sensor on the float. And I also want to use this time to plug a little workshop we have next year at IMAS, the Southern Ocean Biooptics Workshop. Please scan the QR code if you're keen to come along. And I also have a talk at AGU, which isn't related to any of this. It's about ocean fertilization by dust. If you're interested in that, please come along on Friday afternoon. Thank you.
Good morning. I'm Jinfeng Xiao, a research professor from University of New Hampshire. We have been studying the dynamics of terrestrial ecosystems and carbon and water cycles using satellite-derived vegetation indices and solar-induced fluorescence OSIF for many years. So we have been using this data from Landsat, AVHR, MODIS, and the SIF data from OCO2, OCO3, and CHOPAMI. We use them to monitor vegetation phenology. We estimate ecosystem productivity, including crop yield. We look at the impacts of climatic extremes, such as drought, heat waves, and disturbances, such as fire, insect outbreaks, and hurricanes. And we also quantify carbon and water budgets at regional and global scales. Of course, we also use this data to look at the long-term trends of carbon and water fluxes at regional and global scales, as well as water insufficiency. And we also look at how rising CO2, air temperature, and VPD shape all those trends over a long period of time. I was really excited to discover about the availability of PACE data. I'm really interested in using this data to look at the dynamics of terrestrial ecosystems, particularly how useful they are compared to those broadband vegetation indices and SIF data. Yeah, I will be happy to chat with you and develop some potential collaboration. Thank you.
Hello, my name is Michael Taylor. I'm with the Landsat Communications and Public Engagement team. I'm also the Stella team lead. I'm here to talk about Stella. So, you all have been talking about, your various missions, your projects, PACE, Landsat. various other satellites and all that. We're here to try to help people understand what those are, okay? What you're doing, why you're doing it, how you're doing it. And we're doing that through something like this. This is a do-it-yourself handheld spectrometer, low-cost, open source, okay? And we're trying to break down the barriers to make like the data collection, the science, the engineering less intimidating so that anyone, anywhere on Earth can become their own engineer and scientist, okay? Helping build the future, okay? So that anyone who wants one can make their own, all right? So this one, for instance, goes through visible and near infrared, as well as surface temperature, okay, and weather data, and it records it on an SD card. It was made as an analog to Landsat, okay? And so we've had great success with this, all right, from third grade on up for people to understand concepts like spectroscopy and so on and so forth, basically through spectral mysteries. And I can explain more to you back there because I have a nice little booth set up where we're looking at different watercolor, aerosols, and fun things like that as well, okay? So thank you very much. I know it's 2 minutes, trying to keep it short.